The overall goal of this procedure is to measure differentially methylated pre-proinsulin DNA species in human serum as a biomarker of beta cell death.
This method can help answer key questions in the diabetes field, such as, is beta cell death occurring at a given time point during the development of type 1 diabetes? The main advantage of this technique is that by utilizing digital PCR, we can determine the amount of beta cell death in very small amounts of serum.
After collecting serum from human blood samples according to the text protocol, prepare 200 microliters of lysis buffer and 1 microliter of poly-A per sample in a microcentrifuge tube. Vortex the tube for 15 seconds. Remove serum samples from the negative 80 degrees Celsius freezer and thaw them at room temperature. Then, use PBS to bring the serum sample volume up to 200 microliters. Lyse the samples by adding 20 microliters of protease and 200 microliters of the lysis buffer poly-A mix. Vortex the tubes for 8 seconds and incubate the samples at 56 degrees Celsius for 10 minutes. Then, centrifuge the tubes at top speed for 7 seconds. Precipitate the DNA by adding 230 microliters of 100% ethanol to the samples and vortex the tubes for 8 seconds. Then, centrifuge the tubes at top speed for 7 seconds. Next, apply 600 microliters of sample mixtures to spin columns and centrifuge the columns at 6,000 times G for 1 minute.